There's a lot of talk about masks right now, and clearly some skepticism around their use. Now, in general, when it comes to claims that are data-driven, healthy skepticism is a good thing. Questioning and challenging conclusions is a really important part of the process of science. In fact, it's necessary for continually building better scientific understandings. Personally, I wasn't familiar with data surrounding masks, never had any reason to be, and so I was curious about what was out there. So I spent some time reading up, and I thought I'd put together some cliff notes that I could share. I'll focus here on data related to whether masks are able to reduce the spread of COVID-19. Let's take a look at just a couple studies. It's going to be nowhere near an exhaustive list, but I'll try to give at least a few examples of relevant data. The first study was published in 2009 in the journal Emerging Infectious Diseases. I'll put the references in a comment that's pinned to this video in case you want to take a look. In that study, the authors tested the ability of both surgical and N95 masks to impede the transmission of influenza. Remember, this study was published in 2009, so long before COVID-19. The design of the study was pretty simple. They found someone with influenza and asked them to cough 5 times on a petri dish that had some material to capture the virus particles. And then they took a sample from the dish and put it in a tube. We'll call this the pre-mask sample. Next, they had the person put on an N95 mask and do it again. We'll call this one the N95 sample. And then they did it a third time, this time with a surgical mask instead of the N95 mask. We'll call this one the surgical sample. And then finally, they took the mask off and did it unmasked one more time. We'll call that one the post-mask sample. Then they measured the amount of virus in each of the 4 tubes. They did this for 9 different influenza patients. Here's the table of results published in the paper with just a couple edits for clarity. It has 3 main columns. The first is going to contain the data for the pre-mask sample for each individual. The next column has the data that was collected with the masks on, and is actually divided into 2 subcolumns, one for the N95 masks and one for the surgical masks. Finally, we have a column for the post-mask samples. It's not critically important, but in case you were wondering, they used a method called quantitative PCR, we often call it qPCR, to measure the amount of virus in each sample. With qPCR, when a sample can be detected, lower values indicate higher amounts of virus. So, what'd they find? Well, in the trials without a mask, they detected at least some virus in most samples, 7 out of 9 in both the pre-mask and post-mask samples. I should say that the degree of difference in the values can be kind of misleading if you're not familiar with qPCR. For example, the difference in 38 and 40 might seem small, but probably represents differences maybe on the order of 2 to the power of 2 or 4 times the amount of virus in the 38 sample as compared to the sample with a value of 40. And the difference in 27 and 32 might represent something more on the order of 2 to the 5th power or 32 times more virus in the sample with a score of 27. But the more important thing here is how these results compare to those from the masked trials. In those, no virus was detected on the petri dishes from any of the patients. Now, this doesn't necessarily mean there wasn't any virus there, but the qPCR test is one of the more sensitive tests available, so it's safe to say that at worst, the virus made it through to the petri dish in very small amounts, and clearly lower amounts than when no mask was used. So, this is one piece of evidence that's at least consistent with the idea that masks may reduce disease transmission. Let's look at a second study. This one's from 2013 and is titled Testing the Efficacy of Homemade Masks. Would they protect in an influenza pandemic? In it, the researchers took a couple different approaches to assessing the effectiveness of masks. I'll just highlight one part of the study here. Like for the last study, article information is in the comments if you want to take a look. In this study, they were interested in testing different types of mask material to see how well they might filter microbes that cause disease. Again, this was in 2013, long before COVID-19 was on the radar, so they were primarily thinking in the context of influenza. They set up a closed system that air could continuously flow through. I'm sure it didn't look exactly like what I have pictured here, but this should give you an idea of how it worked. The system had a section that was behind a filter, and they did multiple tests with different filter material, including sections cut from surgical masks, t-shirts, pillowcases, and some other things too. Next, they added in some microorganisms that are similar in size to influenza viruses. They let things circulate for a bit, then sampled the air on each side of the filter, and measured the concentration of microbes on each side. From that information, a simple calculation gave them a statistic called the filtration efficiency for each material. This can be thought of as the percent of organisms that got filtered out by the material, with 0% meaning the filter had no effect, and 100% meaning none of the microbes got through. This is a table they reported with their results for each material. There will be two columns of data because they did the experiment for two different organisms, one that's a little smaller than the influenza virus and one that's a little bigger. Let's look at the results for Bacillus atrophius first, which is the bigger one. Each material was actually tested 9 times, and the average or mean filtration efficiency across the 9 trials is what's reported here on the left. So, across the 9 tests with a cotton t-shirt, an average of around 69% of the microbes were filtered out. The column labeled SD gives us a sense of the variability around this 69%. For example, you can think of the standard deviation of 10 for the t-shirts as indicating that the different filtration efficiencies calculated for the 9 independent t-shirt tests generally fell between 59% and 79%. Notice there's a fair amount of variability across materials. The lowest efficiency was silk, which filtered about 58% or a little over half, and the highest efficiency was the surgical mask material at over 96%. How about for the smaller organisms? As you might expect, the efficiencies are generally lower here than for the larger ones, but in most cases at least 50% and sometimes quite a bit higher are still getting filtered. Okay, so that gets us through 2 bits of data around masks. Again, this is far from an exhaustive summary, but the big question still is, if these data do indeed reflect a true reduction in transmission, even if it's not eliminating all transmission, is that likely to have a meaningful effect on the rate that disease spreads? That's actually a little hard to answer here, as it probably requires some more complicated modeling than I want to get into and probably than you want to hear about. But one important part of any of the models we might use will be something you've probably already heard about, the R value. If you're not aware by now, the R value basically represents the number of people a sick person is likely to infect, on average. So if one person gets sick and they infect 2 people, and then each of those 2 people also infect 2 people, the R value would be 2, because everybody infected 2 others. As another example, say 10 people got sick. 9 of them each infected 1 person, and 1 of them infected 2 people. This means a total of 11 people got infected from those original 10, which would be an R value of 11 divided by 10, or 1.1. The reason there's so much interest in the R value is it reflects how quickly the disease spreads. And it's not exactly intuitive, but it turns out that it doesn't take a very big change in R to have a huge change on disease spread. I've tried to put a little graphic together to make this point. I know this just looks like some gray lines on a graph, but it's actually a whole bunch of really small gray points. We can think about them each as representing a person. In fact, what's actually here are 30 rows that each have 20,000 points. Think about each of these rows as dots representing 20,000 people, maybe everyone in a small or medium-sized town. We'll use red dots to show sick people, and we'll trace the number of sick people that might be expected going from day 1, the row on the bottom, to day 30, the row on the top. And we'll look at how different R values impact those expectations. Here's what we might expect with an R value of 1. And here's 1.1, 1.2, 1.3, 1.4, and 1.5. Notice that a difference of 1.2 to 1.5 turns out to have a pretty big effect in those later days. Admittedly, this is a really simplified example, but it does represent the concept of how small changes in R, the growth rate or the infection rate, get amplified exponentially to result in big effects overall. Now, the goal here is not to be alarmist or to bring a message of doom. In fact, you can look at it in just the opposite way. What this suggests is that any behaviors that work to reduce transmission, including wearing masks, while they might seem trivial, actually have at least the potential to make major differences. It's not unreasonable to think that getting that R value, that average number of people that are infected by any one sick person, bringing that down just tenths of a point, can be the difference in hundreds of people getting sick versus tens of thousands, like in the example I just showed, and the effect will likely be even greater when you're talking about larger populations, say on the level of bigger cities or states or countries. And that's the main idea with masks. The data in the 2 studies we looked at here, and lots of others that are broadly consistent with them, suggest that masks at least have potential to slow disease transmission. Hope that's helpful. If so, feel free to share, like, or subscribe to the channel for similar videos.